Yes, thank you for joining today's talk. Will is a postdoctoral researcher at the Water Research Institute at Cardiff University. He's working as part of the data analyst team on the WEWASH project. Will completed his PhD from Bangor University in 2020. He's worked on topics such as Atlantic salmon, aquaculture, gut microbiomes, and biomonitoring using environmental DNA. Now he's focusing his attention on the future of wastewater surveillance. So whenever you're ready, Will, you please take it away. Thank you.
That's brilliant. Thanks, Garmin. Let me share my screen. Fantastic. Yes, great to be here. Hi, everyone. So I'm Will Perry, based at the Water Research Institute at Cardiff University. Like Garmin said, working on the WEWASH project, which is the Wales Environmental Wastewater Analysis and Surveillance for Health project. And today I'm going to be talking to you about from poo to policy, wastewater surveillance in the COVID-19 pandemic and beyond. So I'll jump into it. So what is wastewater surveillance for health? And the main thinking behind wastewater surveillance for health is that when a person has a particular disease, they will produce biological markers in their feces, in their poo, and that poo then will find its way all the way down into the wastewater treatment works, where we can take a sample of that wastewater and look for the particular biological marker that is inherent of that disease. Sadly, the wastewater systems are not this simple. It would make our lives a lot easier if they were, but they are very complex, and so I'll talk a bit more about that later. But in the WEWASH project, we've been looking at SARS CoV-2, and we've been looking at the RNA within the virus in order to detect prevalence within communities. So what is the history of wastewater surveillance? What has been happening before the pandemic? Well, wastewater surveillance is not new to the COVID-19 pandemic, even though that is where it's gained a lot of traction. But it's been going on since the 1940s with, simple cell culture methods for polio being used in the 1940s in the USA. In the 1980s, there was a movement towards cDNA probes, looking for hepatitis A in wastewater in the US as well. In the 1990s, things moved to the polymerase chain reaction, and it's similar methods to what we use today. And, it's really proven its worth even before the pandemic in places such as Sweden, where hepatitis A and the norovirus were detected in wastewater as an early warning system, and allowed policymakers to react to that and prevent widespread transmission of these diseases. And then in 2013 as well, there was a polio detection in the Israeli wastewater monitoring system, and that allowed them to have an early warning system and allowed them to act upon that information that they were receiving early on. And then in 2014, during the World Cup, Brazil also detected polio as well, using their wastewater system in 2 airports. So, it's really proven its worth throughout its recent history, and continues to do so today. Which brings me to Wales in the UK and the wastewater monitoring that has started here. We're behind in some ways. These other countries have been monitoring polio and hepatitis A, but this was really the first widespread program where we were using wastewater to monitor disease. And so in the WEWASH project, we're based at Cardiff University, the Cardiff University Hub, as well as the Bangor University Hub. And we process samples from North and South Wales. We currently sample at 46 wastewater treatment works across Wales, and you can see them in grey there. And there's this north-south split of where those samples are processed and sent to. But, a really good representation across Wales, both in the north and south, where the main population hubs are, as well as sampling in mid Wales as well. And we sample 5 days a week. And so in total, we sample 230 samples a week, and they're all processed on a weekly basis, ready for a Welsh Government report at the end of the week. So who are our stakeholders? So at the front end, we've got people like Cymru, Welsh Water, who operate the wastewater treatment plants and provide water services for the majority of Wales. So in this map here, you can see the catchments of the that come under Welsh Water. But then we've also got Hafren Dyfrdwy as well, the 7D wastewater company that just plugs in that little gap there and services those wastewater treatment plants in that region. And then on the back end, we've got Public Health Wales and the Welsh Government that are interested in the results that we produce. So Bangor University and Cardiff University generating these results for public health. And I wanted to take you through the life cycle of a wastewater sample, because I find it massively staggering just how quickly these samples are processed. Having come from a more traditional academic background where you collect your samples and you can be sitting on them, or sitting on them and the analysis for days, weeks, months, if not years before anything is published and shown to the public, I find it incredible how rapidly these samples are processed, data is analysed, and then results are made available. So I'm using an example from Haverfordwest in far West Wales. That's our furthest site in West Wales. If I go back to the map, that's this one here, I believe. And that sample will be collected on a Monday, for example, although it's sampled 5 days a week, as I said previously, but we're using this one example from the Monday. So that sample will be taken at 12 o'clock, driven to Cardiff University with our ground team. They will transport it along with all the other samples that they've picked up on the route, and it will enter the lab at Monday at 3 o'clock, where it'll go through the first step of the molecular process, which is concentration of the RNA that we want to find within the wastewater. By 12 o'clock on Tuesday then, it's done an overnight concentration step, which I'll talk a bit more about in a minute. And then it goes to RNA extraction, where we extract the RNA from the virus. So SARS is an RNA virus, and we want to extract the RNA within it to find out whether or not it is in that wastewater. So that's by Tuesday, midday. By Tuesday, 5 o'clock, the extraction is complete and it is sent on to qPCR, which is quantitative PCR, the polymerase chain reaction. And this is how we detect the levels within the wastewater, or within our extracted RNA sample, which originated from a wastewater sample. Then overnight, that sample is running. By Wednesday morning, the data is analysed. And by Wednesday, 2 o'clock, that report hits the desk of someone on Welsh Government. And so I just find it absolutely rapid that you go from poo to policy, if you like, in just 50 hours. So really rapid turnaround and a really efficient pipeline. And some amazing people behind this pipeline as well that make it all work. So I'm involved in the data analysis side, where I process the data coming in from all of our different avenues. And it all begins with sample collection, really, at the wastewater treatment plants. And that can happen in one of two ways. So firstly, you've got the grab sampling, which is highly technical, using a stick and a pot. And that pot then gets dunked into the wastewater treatment works to collect that wastewater. And as you can imagine, this way of sampling isn't particularly representative. So it all depends on what's floating past at that particular time that you dunk your pot in. A very simplistic view would be if you had someone who had COVID and they defecated and you had their feces floating along, and you pick that up, then you're going to have a very high signal. But actually, you sample there 5 minutes later and you could have a totally different signal. Of course, the wastewater treatment works is far more complicated than that, and there's lots of mixing, and the catchments for some of these wastewater treatment works are absolutely massive. But that's the idea. It's not particularly representative, but it still does work surprisingly well. And then, of course, you have in this second photo a composite sampler. And so a composite sampler is where the sample is taken over a 24-hour period or even longer, depending on your goals. And it takes little bits of sample making up that 1 litre over a certain period of time. And that is far more representative of what's going on in the wastewater treatment works. So once we've taken our sample, and I must stress that with the Wales programme, there's been a huge investment in these composite samplers, and the majority of our samples are taken via composite samplers now, which is really great, a really great progress. And these are the samples that come in. So you get 1 litre bottles of wastewater. And just by looking at them, you can see that the composition of these samples is radically different between sites. So on the far left, we've got one from Newport. Then moving right, you've got one from Pontier, you've got one from Llanfoist, and then one from Cardiff as well. And so you've got different hues of colour, and in particular, that Newport sample sticks out. And I think this is really representative of the fact that no one wastewater sample is the same. The complexity of these systems is quite staggering, and the inputs that go into them is different depending on different land uses that go into the catchment, sizes of the catchment, of the wastewater treatment catchment, for example. And of course, in the UK and in Wales, we have a combined sewer system where we not only take sewage, such as feces, urine, grey water, such as stuff from your bath or your shower, or if you're cleaning the dishes, all that domestic stuff is going in there. But not only that, you also have things entering from clinical settings, so hospitals, for example. You've got industrial settings or industrial inputs into the system as well. And this is probably what is causing this very discoloured black wastewater here in Newport. So you're getting all that. And on top of that, you're also getting surface runoff as well, so rainfall. So all the rain that hits your roof or the roads, a proportion of that does enter the sewage system, which is something that I'll talk about a bit about later on, but can cause us problems. So we take that wastewater sample and we condense it down, and we do this using a centrifuge, a number of centrifuge steps, as well as polyethylene glycol or PEG precipitation. And what this does is it just precipitates that RNA out of the wastewater that we want to get, or the nucleic acids. It gets those nucleic acids, RNA, DNA, from the sample, and that is what we're interested in predominantly with this project. And it becomes a far more usable volume within the lab setting then as well, in just these little Eppendorf tubes. So it's going from 1 litre to 500 microliters. So that's the first step. You then have RNA extraction or nucleic acid extraction, where you extract RNA and DNA. So for the SARS CoV-2, that is an RNA-based virus, and so we extract the RNA there to try and detect it. And I always find this process rather, miraculous and magic, if you like. And it's a methodology using magnetic beads. And so this is a molecular approach where you've got these magnetic beads in blue, you've got your sample here, and within the sample, you've got red dots, which represent our SARS CoV-2 particles. And then you've also got these other particles, which could be other viruses, it could be other strands of DNA, or just other particles that we're not interested in. And so these magnetic beads are added into the sample, and depending on the concentration of the magnetic beads that you put into the sample, they will bind to a particular size nucleic acid. So a particular size, and here they're binding to our SARS CoV-2 particles, and this is what we use to pull them out. So we use a particular concentration of magnetic beads to pull out our SARS CoV-2 RNA particles. And once those magnetic beads are bound to our SARS CoV-2 particles, you use a magnet, and that magnet then pulls those magnetic beads along with the SARS CoV-2 RNA that are stuck to the beads right to the other side of the tube. And that allows you then to pipette out everything else that you don't want, just leaving you with your RNA and DNA that you're interested in. And I've got a video here just to show you. So we use 96 well plates on a far larger scale. This is, a single Eppendorf, but it's just a representation of what happens. So the beads have been added here to our sample, to the wastewater sample that has been concentrated down, and it's put on this magnetic plate. And what we should be able to see now, sped up, is the beads moving towards the magnetic plate and all being stuck towards that side. And it's, it really is like a magic trick, I think. Those beads move to the magnetic plates, and with it are the RNA molecules that we're interested in. So our SARS-CoV-2 is all there in that little dark splodge, which is great. So we take our extracted RNA from our SARS-CoV-2, and we look at a number of regions within the genome of the SARS-CoV-2. So we look for particular markers, and these are the E gene, the N1 gene, and the N2 gene. So the E gene is used in the production of the envelope proteins with the virus, and the N genes are used for the nucleocaspid proteins. And so that's their function, but we use them as markers to look for the SARS-CoV-2 virus. And we also look for another virus, which is crasphage. So this is a bacteriophage that is found within all of our guts. It preys on bacteria, and so it is found in feces, in poo, and we use the crasphage to give us an idea of how much poo there is in the sample, because they're so closely linked. And the process that we use to look for our crasphage and our SARS-CoV-2 using these different gene regions is a process called quantitative PCR, or the polymerase chain reaction, where we add probes that look for these gene regions and look for our crasphage, and it amplifies them. And depending on the amplification and the fluorescence that the probes give us from the amplification, that's what's all going on inside this machine, we get an idea of just how much of the SARS-CoV-2 we have, or just how much of the crasphage we have. And so here you can see different amplification curves for our different markers, and also for different samples. So where there has been a positive result for SARS, for example, we see an amplification here going across. And using these plots, we can then work out just how many gene copies there are per liter of wastewater. Which brings me to the data analysis. And the main body of the data analysis is to do with normalization. And we normalize for two things. So we normalize for dilution, as well as the number of people contributing to the wastewater in a particular catchment. So you can imagine that if it has been particularly rainy, our sample is going to have higher dilution. And if you can imagine, then if it's more dilute, you're going to pick up less SARS-CoV-2. And perhaps, it's really important to account for this normalization, because if we see a reduction in SARS-CoV-2 in the sewers, in the wastewater system, it could be that it's rained rather than the amount in the community has gone down. We also normalize for the number of people. So, for example, Cardiff is an absolutely massive catchment, wastewater catchment, and we are comparing them with other catchments that are far smaller. So Bulth Wells, which will be, very small compared to the Cardiff catchment. And so we're going to see less SARS in the wastewater because it's servicing less people. And so these things are really important to take into account. And so what we did originally is we took our SARS CoV-2 levels, so the gene copies per litre from the qPCR machine, and we divided it by the amount of CRAS phage that we were seeing. So again, gene copies per litre of wastewater of CRAS phage. And so we did a SARS divided by CRAS phage, which gave us a normalized SARS signal. Because like I was saying, CRAS phage is found in most human intestines and feces. And so it gives us an idea of just how much human feces there is in the wastewater. And that will then take into account for dilution, because if it has rained, the SARS virus will be more diluted. And so we know then it will work as a dilution factor for our SARS-CoV-2. And it's also representative of the number of people as well. So the more human waste, the more people, the more CRAS phage you're going to find. And so this is what we use to begin with to normalize our signal. And yeah, so it's both a type of biological marker. But then we started to see some issues. And so this was back in October 2021. And you can see our normalized SARS signal here based on the methodology I've just mentioned. And we were seeing peaks and troughs, which we weren't expecting, and which were of much higher magnitudes than what we'd seen before. And if you cast your minds back to October 2021, we saw a vast amount of rainfall. So here is the Met Office October 2021 rainfall amount percentage of the average between 1981 and 2010. And you can see in South Wales here, a lot of places had above average rainfall, and in North Wales as well, really high levels of rainfall for October. And like I mentioned before, we've got a combined sewer network. And so that water, or, a lot of water then from rainfall is entering the sewage network, causing a vast amount of dilution, which should be accounted for by the CRAS phage virus, yes. But the other problem is that the sewer network becomes far more complicated when there's more stuff in it and when it's raining. So this is a map of the CSOs, so the combined sewage overflow, which I'm sure you've heard all about on the news, which are really rising to predominance and public interest at the moment. And we can see Cardiff down here, and just the number of CSOs that are spilling is large. With each one of these CSOs, so I should say a CSO spills when the wastewater treatment, or when the wastewater system is overloaded. So when there's too much volume in the wastewater system, mainly caused by rain, then these CSOs are opened to stop sewage flooding into homes and businesses and all that sort of stuff, and from overwhelming the network. And so lots of CSOs going around South Wales. Well, it's a national problem. But with those CSOs, you lose wastewater into the natural environment. So those CSOs then spill into rivers and into the ocean, less so into the ocean in Wales, because we've got things called storm tanks. But we're losing that sewage into, or that wastewater into the natural environment, and it doesn't go to the wastewater treatment plants. And so we cannot monitor that. And so we're losing signal there, which is one issue. The second issue is that when it's raining and the wastewater system is overloaded with large volumes that it normally doesn't have to cope with, water is pumped into these huge storm tanks. And again, this is to stop the wastewater being pumped into the environment and being put into beaches and rivers. And so, yeah, I didn't realize just how big that these tanks were, but here's a photo with some people for scale. So large volumes of wastewater stored in these tanks in times of high volume, and then that water is pumped back into the system later on when the system has the capacity to deal with it. But the problem with this is, when we've got all that wastewater there, it can be pumped back into the system, days, perhaps even weeks later. And so all of that SARS signal and CRAS phage signal that we see in that storage tank is then pumped back in later on, which is an issue. And the fact that we were using CRAS phage, which is a DNA-based virus, and we were also looking at SARS, which is an RNA-based virus, the SARS is going to degrade quicker than the CRAS phage. And so if you can imagine a setting where you've got a SARS signal decreasing more rapidly than a CRAS phage signal here, then that normalization process starts to break down using the CRAS phage biological marker. And so instead, we move to a flow and population-based estimates for normalization. So like I said earlier, we normalize for dilution and we normalize for the amount of people that are contributing to the wastewater. So rather than using CRAS phage for the silver bullet to normalize for both of these things, we use flow into the wastewater treatment works as an idea of how dilute the sample is, and then population estimates to get an idea of how many people are contributing to that wastewater treatment works. But then another issue arises in that the wastewater treatment system is highly complex and it differs between different wastewater treatment works. So this is a schematic of a wastewater treatment works, and they measure flow. So Welsh Water measure flow at different stages at different sites. So some sites they measure flow at the influent, some sites they measure flow at effluent, so how much is going in and how much is going out, and then some sites they'll measure flow somewhere in between. And the issue with this is that those readings, depending on where you take the flow, can give different estimates. So we've got some examples here where we've got effluent in purple. So that's the amount of water coming out of the wastewater treatment works. You've got influent in like a turquoise there, which is the amount of wastewater coming into the wastewater treatment works. And then you've got near inlet, which is measured somewhere within the wastewater treatment works. And these are from 3 different sites that are very close to each other. So 3 different sites measuring flow in different ways. And what we can see is that you can see peaks in flow for influent, but we start to see these plateaus for effluent and near inlet. And this is because, so the gold standard is the influent, and that will tell you how much wastewater or the flow of wastewater going into the wastewater treatment works. But things can happen within the wastewater treatment works that causes these plateaus. So we should see peaks here as well. But because wastewater is pumped into storage tanks, it's released through CSOs, we see a plateau. So this is the maximum capacity for that wastewater treatment works. And so we don't see the flow go any higher because the rest of the flow, which should be coming up here, is being diverted elsewhere. And so we're not getting an accurate representation of flow for all of the sites that are using the effluent flow and near inlet flow. So the way that we got around this is we use proxy flow, and proxy flow is based on electrical conductivity, ammonia, orthophosphate. So these are the chemical parameters that we also measure alongside our biological markers. And then we also use CRAS phage, and we use these markers to work out just what the flow should be. So we model a proxy flow based on these measurements. So you can imagine here when we see the plateau, using our electrical conductivity in ammonia, orthophosphate, and CRAS phage, we'll start to see dilution in these measures, even though we don't see any difference in the flow. And so that is how we model a proxy flow and build in flow that we're missing here. So that's how we've got around that, and that is what we are currently using for our reporting. Which brings me on to the reports, and this is one of our older reports that we were producing for Welsh Government. Here you can see our dodgy area, and we were looking at South Wales signal across, so we got mean normalised SARS. We were also looking at the North Wales signal. I've not included that in here just for brevity. We were looking at the amount of SARS in the wastewater compared to the case numbers, which is in red. We've stopped doing that so much now because the case numbers are becoming more and more meaningless as people stop testing. And we also, had the breakdown of different sites as well to see what was going on. So this is just, again, some North Wales sites that I've picked, just to keep it brief. So these are the sort of reports that we were handing to Welsh Government for their decision-making process. And you can find these reports here at Welsh Gov, the COVID-19 situational reports. They were going into the situational reports alongside case data, prevalence in hospitals, prevalence in care homes, all of those pieces of data alongside with the wastewater data that we were producing were being given to Welsh Government for decision making. We've now moved to a new reporting type, learning from our mistakes or learning from the difficulties that we've had in the past. So we now use that proxy flow based normalization, which is working great alongside the normalization for population. And we've got some nice heat maps in there now as well. This is just the all Wales SARS normalized signal. And we got the different regions there in grey as well. And this has now turned into a 52-page report that sits in its own, its own independent report, as well as being included, a summary of it being included in the situational reports given to Welsh Government. And so now we have the space to give breakdowns on each particular region, whether or not we're seeing, rapid increases or if we're seeing rapid increases or declines over long periods of time. These things are all picked up in our indicators for each of the different regions. So here I'll just show the Cardiff region. So we've got 2 wastewater treatment plants or catchments that are contributing to the Cardiff region, which is Kilfynydd and Cardiff Bay. So I've gone through who to policy, or what we give the policymakers so that they can use that in their policy making decisions. And it leaves me really to sit on the benefits. So what are the benefits of wastewater surveillance for health? Well, it's cost effective in terms of monitoring. You don't have to spend lots of money giving each person in a population a test. You can capture large portions of the population with the wastewater treatment works and just test in that way. So it also allows for early detection. If you've got these systems in place already, they're non-expensive compared to testing everyone in the population. And so you can have them operating when you are not in a pandemic. And so you can, like we've seen in those examples I gave at the start of the presentation, in Israel, in Sweden, in Brazil, they've all been used as successful early warning detection systems. You're testing asymptomatic and symptomatic people. So not only people who have symptoms who are getting tested, you are testing everyone because everyone has to poo and everyone has to use the sewage network. It also allows for a huge amount of automation. So everything from the concentration stage in the lab onwards is highly automated, which means that we can process the large number of samples, the 230 samples a week that we're processing currently. And it means that we can ramp up that sampling as well relatively easily. It's non-discriminatory, like I said, in terms of asymptomatic or symptomatic, but also in terms of societally who is going for tests. That doesn't matter because you are testing everyone who uses the wastewater system. It's independent of health-seeking behaviours. So this was particularly important at Christmas time, where people weren't getting tested because they want to see family members and other holiday periods. But everyone has to poo, and so you will pick up that signal in the wastewater. It's independent of clinical testing, and it's also customizable, which brings me on to my next point, which is post-pandemic. What is the role of these wastewater surveillance systems post-pandemic? So it's very easy to change modules within this pipeline. So for example, you could change the markers that you're using for your qPCR. So currently we're looking for SARS CoV-2 and we're looking for crassphage, but we could use different markers to look for different viruses, which would be very easy to implement. Or you could even remove that section entirely and remove that module and place another lab module in there to look for a whole host of other different things. And that is really where your imagination can run wild, and where it gives you the true flexibility and customizability of the wastewater surveillance, because this lab-based setting can be swapped out to look for other things. It can look for other viruses. So norovirus, perhaps, other emerging pandemics. It can look for those viruses. We can look for antimicrobial resistance. We can look for chemical markers related to drugs, be they illegal or just the drugs that people use on a day-to-day basis in a healthcare setting. And even, looking for biological markers related to cancers, so bowel cancer, perhaps. Would that be a useful avenue to look at cancer rates? So really, there's a huge amount to explore with wastewater, and there's a huge amount of value in that wastewater. And I think you can go even one step further. So you can change your markers. You can look at DNA extraction rather than RNA extraction. And you can even change the samples that are going into this system, into this pipeline as well. And so, could we see a role for these surveillance going outside of wastewater and just wastewater surveillance in general? Just water surveillance, not wastewater surveillance. So going from these lovely channels of wastewater and sewage to a river, taking samples instead from rivers around Wales rather than wastewater treatment works around Wales. And rather than looking for viruses such as SARS CoV-2 or crassphage, could we look for eDNA? So DNA released from organisms into the river. And very quickly, you've got a biomonitoring system rather than wastewater monitoring for healthcare. You've got a biomonitoring system for wildlife and biodiversity. So is that a possible role for these pipelines and for these types of water monitoring systems after the pandemic? Which is also being talked later on by in the series by Dr. Lindsay Harper, who will be talking about environmental DNA and is from Nature Metrics, which are seeing a massive surge in numbers and popularity, and they're really flourishing as a company. And so, is there a role for these water monitoring systems moving from wastewater just to water monitoring systems on a national scale, again, for government, perhaps? That is a possibility. So what are the roadblocks that really stop the progression and setting up of wastewater or water monitoring programs like this? And I suppose you've got to have your molecular techniques honed. You've got to know that they work and that they're reliable, especially if that's being used to inform policy. It's getting the stakeholders together, so making sure, for instance, in the WEWASH program, making sure you've got Dŵr Cymru and the water utilities on side, and then also making sure that your results are being sent in a timely fashion to policymakers like Welsh Gov and Public Health Wales. So it's getting the stakeholders together and getting them right. It's setting up that laboratory infrastructure that allows you to take samples, be it a biological marker or chemical marker, and being able to process them rapidly or in a time frame that is useful for policy. And then it's also having the personnel, so the people to do it, from the people who take the samples, from the people who are in the lab processing the samples, the people who analyze the data. You've got to have the right people there to make sure that that runs smoothly and that the data is produced, analyzed and reported in a timely fashion. And then of course, there's also, you've got to have the appetite to do it, and there's got to be the means to be able to do this, which perhaps is maybe one of the largest roadblocks currently. So I'll leave it there on the post-pandemic thoughts and what we're going to do after the pandemic. Hopefully it ends soon and we move to an endemic state. It is going that way. But yeah, hopefully I've highlighted how useful the wastewater monitoring programs are in terms of health with WEWASH and all the amazing stuff that is being done behind the scenes, but also the other things that these wastewater programs have the potential to do as well, and maybe even moving on to water surveillance rather than just wastewater surveillance. So that leaves me to say thank you to all my colleagues at Cardiff University. There are so many to mention. I couldn't list them all, as well as colleagues at Bangor University as well. You know, just so many people go into producing this this report and the data that goes into these reports. It's really, really brilliant, and it's a well-oiled machine. And then, of course, thanks to Duo Cymru, Welsh Water, Hafryn Dyfrdwy, Public Health Wales, and our colleagues at Welsh Government as well, who we're producing this data for. So I'll leave it there and just say thank you for listening, and I'll take any questions.